Today, the Royal Tyrrell Museum and its cooperating society are proud to present our very own Dr. Don Bregman, who is also the Director of Preservation and Research at the museum. Don is originally from Craigmile, Alberta. Growing up in southern Alberta, he developed a passion for paleontology at an early age while collecting fossils in the Alberta Badlands. Wanting to turn his passion into a career, Don decided to enroll in the paleontology program at the University of Alberta. After completing his bachelor's degree, Don moved to Montreal to pursue his PhD at McGill University, where he studied the structure and function of the ankle joint in diapsid reptiles. Upon completing his PhD, Don was hired by the Museum of Comparative Zoology at Harvard University in Boston, Massachusetts, to help upgrade the collections. This work involved identifying and cataloging a large number of fossils collected in Permian rocks from Texas. It is during the final year of this project that the Royal Tyrrell Museum was established and that Don was asked to join the ranks of the resident paleontologists. Don's research interests focuses on two main areas: the taxonomy, biostratigraphy, and distribution of Mesozoic turtles, as well as the use of Cretaceous vertebrate microfossil assemblages to study paleoecology and vertebrate distribution. Over the years, he has undertaken fieldwork in Canada, China, Mexico, Argentina, and the USA. Today, Don will talk about the diversity of North American fishes at the end of the age of dinosaurs. So without further delay, here's Dr. Don Bregman.
Thank you, Wendy. I was looking for you to read the title. Somebody pointed out it's the longest title in the series. I was trying to be precise, and then as I went through the talk yesterday, I realized that it's not correct because I'm also going to include reference to elasmobranchs as well as teleosts. But just to give an overview of what I want to do in this talk, I want to present work I've done on the fish from microvertebrate sites. I'll start with an overview work I did on Dinosaur Park, trying to understand the fish assemblages from there. And once I had that, the big question that emerged was how do these fish assemblages change through time and space? And I was able to study material from a variety of times, and we'll present the results of studies from the base of the late Cretaceous up to the time of Dinosaur Park based on material from Utah and Alberta, and then going from the time of Dinosaur Park to the very end of the Cretaceous. And the final part will be looking at the changes in fish across the Cretaceous tertiary boundary. So just to give a bit of introduction, I think everybody's really keenly aware that Dinosaur Park is really an exceptional locality, and it's been crucial for the museum and the history of the museum. When I was first interviewed by the museum, it was pointed out that the priority that they wanted us to undertake in terms of research at that time was looking at the paleoecology, looking at how animals lived, interacted, and how the communities were structured. Dinosaur Park is an excellent place for this because of its exquisite fossil record. The dinosaurs are obviously central to the museum and provide a very high profile to the assemblage from Dinosaur Park. The studies of paleoecology of the dinosaurs have really, based on Dinosaur Park, has really vastly extended our understanding of dinosaur paleoecology of the late Cretaceous, and are the basis for how much of this information is presented in the gallery. But at the time, it was a very wet environment. This is a reconstruction that Dave Eberth did of his concept of what the Dinosaur Park formation would have been like. Big rivers, lots of standing water, and in this water there would have been communities of vertebrates that would have lived in the water. I've spent a lot of time looking at the mesoreptiles, the turtles, crocodiles, and champsosaurs For these, the record is very good. There's lots of articulated specimens for most of the taxa that occur there. There's a few that are not, but by and large, most of the taxa that occur in the Dinosaur Park formation of aquatic reptiles are represented by articulated specimens. When it comes to fish, it's a totally different story. This is pretty much our full record of articulated small-bodied fish from the Dinosaur Park formation. There's a few larger things, but for the small fish from the assemblage, there's one skeleton described as Cretoferiotis and a brain case of a fish called Balastomus, a long-bodied brain case. This really presents a challenge to understanding the assemblage because most work on fish is based on articulated skeletons. What there is in the park is vertebrate microfossil localities that are where there's lots and lots of isolated elements that are mixed together, including teeth of dinosaurs as well as remains of fish. The way of collecting this material to get large sample sizes involves digging out bags of the rock, soaking it and screening it, and then picking through the residue under the microscope. The results is this kind of material. Lots of isolated elements. This relatively small sample size has at least 5 kinds of aquatic vertebrates represented, so that because of the large number and the diversity, we can take statistical approaches to documenting the communities and looking at patterns of distribution. The challenge is to identify this material. So for some groups, like the elasmobranchs, there's a long tradition of working on taxonomy from teeth especially, teeth and a few other elements, so that from the material from microvertebrates, we know that there's 3 basic kinds of elasmobranchs: a fairly large-bodied shark called Hybotis, Mylodaphus, a guitar fish, and then a fish, or electrolobid, like a carpet shark, is a modern member of that family. For the bony fish, the work that was done earlier recognized a number of basal taxa, including sturgeon and paddlefish, gar, amiids, and an extinct group called Balastomus. The work I did on microvertebrate sites, it soon recognized that there's a few other of these basal fish present in the assemblages, represented by primarily scales and skull bones. These are morphologically distinct, but we didn't know what kind of fish they're related to, so I gave them letter designation. In this case, referred to it as Holostean A, and another one with a distinctive scale with a tuberculate sculpture on the outside, referred to as Holostean B. So adding these to assemblage, these are the basal fish that we've got. And typically people think of this as the fish assemblage from the late Cretaceous. But it's long been recognized that there are teleosts. Teleosts are the common bony fish you think of when you think of fish, the pike, the pickerel, the goldeye. They're the most abundant vertebrate group around today. There's something like 32,000 kinds of fish, teleost fish, that are recognized today. I believe it's 64 kinds present in Alberta. So it's very important in modern ecosystems. And part of the question is, how did this diversity arise? What were the teleosts like at the time at Dinosaur Park? It was recognized by people working on microvertebrate sites that there is diversity there, but because you're dealing with isolated elements, it's hard to sort out. Some taxa were recognized based on jaw elements, tooth-bearing elements. For example, these distinctive teeth and tooth plates were recognized as from something related to the modern bony tongue, albula. And one would think that the best approach to trying to work out diversity is use jaw elements, because with jaws there's a limited number for taxa. Jaws give good indication of something about what kind of feeding style it had and so on. And looking at the material from isolated elements from microvertebrate sites, there are lots of different kinds of jaws, and some of these can be identified. But in looking at the material, I found that actually more informative were the vertebrae, the centra from the backbone. There's a lot of morphological variation and a lot of specimens. The challenge with working with vertebrae is that you've got a large number per individual, and that there's variation from front to back along the column. So there's a lot of kinds of variation that have to be taken into account in trying to sort out how many different kinds of taxa there are present. My approach was to start with a basic morphotype approach, defining morphologically distinctive types of vertebrae, and then looking at those and questioning whether or not those are taxonomically distinct. So for example, this is one morphotype I recognized, very simple spool without processes going off the side and with large openings for the rib articular pits and the neural arch articular pits. In contrast, this is another one where there's processes sticking out the side and distinctive pattern of pits. So taking all this approach, we end up with a number of different kinds of morphotypes. The question then is, what do these mean taxonomically? And to answer this question, I took three approaches. The first one was starting with where we know a taxa is present based on jaws. What are the vertebrae that are most likely from that same kind of fish? So for example, we know that there's an albulid fish based on the tooth plates I showed, looking at the recent vertebral column skeleton of albulids, and then comparing with the fossils, we can identify that as an albulid vertebral column. And since they're occurring in the same general pattern of distribution as the jaws, we're fairly confident in saying these are the centra of Paralbula, the taxon that was named on the basis of the jaws. Another one that we recognized fairly early on was an Esocoid, a primitive relative of the pike, based on features of the tooth implantation. That's most clear here. They've got a C-shaped base of the teeth. And this is because the teeth in pike, if you've ever handled one, actually fold backwards. They're only attached at one side, and the teeth will fold back, and they leave this distinctive feature in the bone. So when we see this, we're confident we've got something that's related to a pike. Looking at the skeleton of a pike, we can say that these centra are of the kind that would be from the kinds of fish that have the pike tooth plates. The problem is there's actually variation in terms of the jaws. There's more than one type of pike. There's more than one type of centra morphotype. We don't know which is which, but we can say this group of 4 different types of morphotypes are pikes, and this group of 4 different kinds of jaws are also pike. One of the really interesting examples that came out was a fish that is a member of a group that's present today in dominated in South America. It's the crassiforms, best known as the tetras, the little aquarium fish you often see. One of the really well-known members of this group is the piranha. They have a very distinctive hinge type joint at the symphysis of the lower jaw. This is the Dinosaur Park example, and that's the modern example. So based on this feature, we're identifying a crassiform in the Dinosaur Park formation. And again, by comparing with the recent skeletons, this is from a recent crassiform. This is the fossil that we're saying is from the same kind of fish as the jaw. We're able to identify the vertebral elements that are crassiform, and then use those to look at distribution patterns. So that was the first vertebrae along the vertebral column, and these are ones from more posteriorly along the vertebral column that we're attributing this type of fish. The second approach to identifying the centra is looking at features that are unique to the group. I mentioned, for example, features that are unique to jaws that allow us to say that one fish is a crassiform. There's also examples of some fish that have distinctive features unique to that fish, and one of them is the hyodontids, the goldeye and mooneye. This is the first vertebrae along the column of a goldeye, And this is a fossil example that we're putting in the same group based on this sort of clover leaf shaped anterior articular surface. So based on this, we can say we've got goldeye in the Dinosaur Park formation. We don't have jaws that we can attribute to this yet, but based on the vertebrae, we can say that type of fish is there. Another one that proved to be very interesting is a fish that's related to the catfish. It's represented by very flat centra that correspond to the first vertebrae along the vertebral column of catfish. There's some features that are not that are present in catfish that aren't present in this Cretaceous example. So it's not a member of the crown group, the living group of catfish, but we were arguing that it's a relative of it, an early member of that lineage. And these are other elements that we've attributed to this group. The first vertebral column, vertebrae from further posterior along the back, and lower jaws. And this is all from a combination of comparing with recent material plus looking at distribution patterns. This leaves a number of vertebral morphotypes that we can't put in extant groups. They're morphologically distinct, but we don't know what kind of fish they're from. To try and resolve this, then the question is, are these taxonomically distinct, or are these regional variations along the vertebral column? To answer that column, I've been relying on distribution patterns, with the idea if they're from the same kind of fish, they should have the same kind of distribution pattern. An example of that is these centra here, all variants along of the Asacoid type centra. The variation is pretty subtle. Just for example, a single ridge here versus a broader bar there. We think that this one I've designated NVE versus NVC are distinct because in one part of the Hell Creek Formation you get NVC but not NVE, whereas in the other part you get both. So because they have the same different kind of distribution patterns, I'm arguing they're distinct kinds of fish and then treating them as distinct kinds of fish in looking at diversity patterns. So that's how we're dealing with the taxonomy. When we put it all together and try and do a visual representation of what we see in terms of the fish in the Dinosaur Park assemblage, this is sort of what we see. All the basal fish, like the sturgeon, the elasmobranchs, the paddlefish. There's a few large teleosts like coreops and paratarpon, but there's lots of fish, teleosts, and they're all very small size. They'd be basically less than 10 centimeters long based on the size of the centra and the size of the jaws. So a very interesting assemblage in that very high diversity of teleosts, but dominated by fish of very small size. With that sort of understanding of the community of fish from Dinosaur Park, there's a number of questions we can ask, and one of them is distribution patterns. Because we can put some of these in extant groups, we can start looking at distribution patterns and recognize connections between different areas. One of the interesting ones is the presence of a hyodontid, this goldeye, in the late Cretaceous in North America. It's thought somehow to be related to a fish called Lycoptera from Asia, which occurs much earlier in time. For a while they were put in the same family. Now it's thought they're related, but the Lycoptera is an earlier representative, and the larger group originated in Asia. But because they're all freshwater, the implication is that there's a fully non-marine connection between Asia and North America, at least by the time of first occurrence of the hyodontids in North America. We know there's a connection because of the dinosaurs, but dinosaurs could potentially swim across straits of marine water, while these fish, which are obligatory freshwater based on what we know of the modern relatives, plus the distribution of the fossils, there must have been a fully freshwater route for them to pass between these two areas. Another example of this is this scale that I designated as Holostean B. It's present in Dinosaur Park in the late Campanian, but it's also present at a much earlier time in Asia and in the high Arctic. So again, this is a fish that's only found in freshwater sediments. All the places it's found are freshwater. So it implies that there was a freshwater connection by this earlier time, the first time of occurrence in North America, the late Turonian. This presence of a crassiform, in this South American group in North America, is also indicating some kind of connection. Here it's much more difficult because to know exactly where, because there is similarities of the South American assemblage all through Africa and Asia. So it may have been via Europe sorry, not Asia, Europe. So it may have been via Europe into North America rather than South America into North America. But somehow at this time, there was some introduction of South American taxa into North America. Again, we know there was an exchange based on dinosaurs because there's hadrosaurs in South America in the late Cretaceous, and all indications are they must have got there from North America. The presence of this fish is adding to the time and also indicating that whatever this route was, it involved a fully freshwater possibility of exchange. So with that sort of look at the Dinosaur Park assemblage, the next questions that came up were how did it change through time? And fortunately, I was able to study material that had been collected from Utah. A colleague, Jeff Eaton, has been collecting microvertebrate sites in Utah for many years and has a sequence going from the base of the late Cretaceous up to the time of Dinosaur Park. I was able to look at this material and look at the changes in diversity going through this time. And just to jump ahead to the pattern, when you identify all the fish using the approach I've used for Dinosaur Park and plot it out, this is the pattern of change you get. A gradual increase with a few jumps where there's major changes. One of these jumps is in the difference between the Cenomanian and the Turonian. It's within the late Cretaceous, but very early in the late Cretaceous. There's 4 last occurrences, 3 first occurrences. There's also changes in relative abundance. The dominant fish in the early Cretaceous and in the Cenomanian are one called something like Lepidotes and a pycnodont, one with angel-like fish body form with crushing teeth called Coleotis. Both of these are easily recognizable by the teeth, and we can estimate their abundance. Really the dominant fish, looking at identifiable elements, about over 50% of the elements in that time are Lepidotes, and something like 20 are this Coleotis, this tycnodont. At the Turonian, they carry on, but they're down to less than 5% of the assemblage. So again, a big change in the assemblages based on relative abundance of these fish. The things that come in, an amide called Melvius, which is again becomes really abundant throughout the Utah assemblage in younger localities. And this is the first time of gar in North America. The gar fish, like the ones we have in the gallery, we think of as typically late Cretaceous, but in North America they first appear at this Turonian record. They're present in South America in earlier beds, so indicating somehow this change may have been an introduction of South American taxa into North America. And then this unknown fish that I believe is somehow related to catfish first occurs in North America in this Turonian assemblage. With this Turonian assemblage first developing, things basically there's gradual changes. A few things get introduced. Some of these really become important later on. So at this second period of change, a couple of the fish that first occur is a second kind of gar that we don't know too much about other than its teeth, but significantly acanthomorphs. Acanthomorphs are the derived teleosts. the ones that, the group that includes things like perch and a number of fish that are common today. And when we look at the record, they're very rare in this, early locality, but they gradually increase dominance in terms of number of specimens in an assemblage, and really take over and become the dominant fish by the into the Paleocene. So evolution in action. The other big period of change is going into the Dinosaur Park formation. And this is interesting because somehow we assume that Dinosaur Park is typical for the entire late Cretaceous, but it's not. There's things that we think of as typical for Dinosaur Park that first occur at about that time. And things like the bonefish and Belanostomus, as well as paddlefish and sturgeon. The sturgeon have relatives in Asia, but in terms of North America, their first record in non-marine sediments is in the time of the Dinosaur Park formation. And interesting also, the Asacoids, the pike, first occur in Dinosaur Park. So a big change there. And because these are new groups that are appearing without obvious antecedents in earlier assemblage, it suggests that there's some kind of immigration event with taxa moving into this area of North America from somewhere else. And that leads to questions of biogeography. What were the distribution patterns? We can test this to some extent by comparing what we find in Alberta to what we find in Utah at the same beds. There's a Dinosaur Park formation in Alberta is the same time as the Kaparowitz formation in Utah, and geographically different. The Dinosaur Park formation we interpreted as having been deposited about 58 degrees north, so just a little bit south of the border with the Northwest Territories, while the Utah was somewhere in the 40s degrees paleolatitude. And some of these taxa that first occur are restricted to Utah. They don't extend at least into Dinosaur Park. And others, like the sturgeon and Belanostomus, are present in Dinosaur Park, but not in Utah. So there is evidence from this that there's latitudinal patterns that are present. Another example are these basal fish that I designated Holostein A, Holostein B. I was always concerned when I was recognizing them as to, are these something that we get that don't occur elsewhere? Is that why they hadn't been recognized? Or are they present but just not recognized because they didn't have a name? And when I was able to look at assemblages, it was true that their absence in Utah and present but very, very rare in Montana. And again, by looking at the geographic patterns over time, it gives us biogeographic information. Because they occur in the Dinosaur Park formation, but they're in North America, at least by the Turonian, it implies that these latitudinal patterns are present over long periods of time. There's also a change in relative abundance that we can estimate by looking at the relative abundance of centra. So just plotting out the relative abundance of two of the types of fish, the one called Coriops in blue and the one called UBVD3 BVD in this crimson-like color. You can see there's a really difference in abundance of these types of fish compared Utah compared to Alberta, and that there's a disjunct distribution in that they do occur in early beds in Utah, but are absent in younger beds in Utah. When we plot relative abundance, we see that this disjunct distribution is reflected in a distribution pattern where they're most abundant in Utah and they drop off in the younger beds with an inverse relationship. This is of interest because when you compare this to the temperature curves, it was high temperature in the time when the southern taxa was abundant and lower temperature when it's rare. So that this is indicating that there's latitudinal patterns, and these latitudinal patterns are changing in respect to climate change. So that sort of gives an indication of big picture changes that are happening leading up to the time of Dinosaur Park. The next question is, what happens after Dinosaur Park? And we can look at this by looking at localities, especially from Montana, from the Hell Creek Formation. These have been intensively studied by people looking at mammals, and large sample sizes are present. They're in the very latest Cretaceous, just before the time of the end of the Cretaceous. Looking at these, there's a few things that appear that don't occur in Dinosaur Park. There's fish Priscacara that's well known in the Eocene Green River Formation that we're recognizing based on isolated elements from the Hell Creek that doesn't occur in Dinosaur Park. And also there's a hyodontid that gets really big. The hyodontid in Dinosaur Park, the centra are a couple of millimeters long. So the fish itself was probably less than 10 centimeters long. This millimeter scale, you can see we're dealing with a centra that's almost a centimeter in diameter here. So a big fish, one that would make a meal. When we plot out just basic numbers of taxa that were recognized based on the studies of vertebrate for the teleost, it appears there's a decrease in diversity. There's 30 different kinds from Dinosaur Park and 20 from the Hell Creek Formation, 21. So that it looks like there's a drop in diversity, but there's a definite sampling bias here because I've looked at Dinosaur Park much more intensely than the Hell Creek has been looked at in terms of sampling from a wide variety of localities. Another way of looking at that is sort of averaging the number of taxa you get in a locality. And when we do that in the Dinosaur Park Formation, we're getting about 9 taxa per locality. We're getting a larger total number of taxa because we're sampling more environments. We've got quiet water environments, we've got stream type environments, we've got environments that are very in a coastal setting. So because of all this difference in environments, we're getting different habitats represented and a different higher number total diversity. But when you average the number of diversity per locality, it's quite low. Whereas in the Hell Creek, it's 13 taxa per locality. So it seems that in terms of the communities themselves, the communities, aquatic communities, are getting more complex, more taxa typically present. Interestingly, this pattern of acanthamorph increase holds, but occurs within the Hell Creek Formation. The lower Hell Creek, the diversity of acanthamorphs, or the abundance of acanthamorphs, is the same as Dinosaur Park, roughly. And there's a jump leading into the upper Hell Creek, so that the changes are they're stepwise, and they occur, in this case, within the Hell Creek Formation. The other big change in terms of the complexity is you're getting more teleosts of larger size. I mentioned the big hyodontid, and this is just a graph indicating the size of centra present in the assemblage, so that for centra above 3 millimeters in diameter in Dinosaur Park, there's only 2, where there's 1, 2, 3, 4, 5, 6, 7 in the Hell Creek Formation. So that the teleosts are basically moving higher up into the community food web, and more of the large-bodied predators are teleosts. So again, the community is becoming more complex. And if we just do the same kind of representation of the diversity of fish and the diversity of fish in different feeding guilds, this is what we get. In the Hell Creek, again, more large-body teleosts, hyodontid, coriops, a large acanthamorph, but still lots of diversity of small kinds. So basically similar structure, but more taxa involved in a community, and teleosts moving higher up into on the food web. So the final question I wanted to look at is, taking the same approach, what are the changes in fish assemblages going across the Cretaceous tertiary boundary? And for this, again, I relied primarily on data from Montana, sites that have been studied, especially by people from Utah, but also a site from Saskatchewan that Corey Redmond is studying intensely, the Pine Cree locality. This is one of the classic localities, the Bug Creek locality, and this is Bill Clemens, who is one of the leaders in study of microvertebrate mammal assemblage, collecting of mammals from the late Cretaceous and Paleocene in Montana, and his student Greg Wilson at the Bug Creek locality, which Dennis and I were fortunately be able to visit and get the story of the locality firsthand. A few new things appear, and one of the striking ones is Esox itself. I talked about the Esocoid that we see in Dinosaur Park. It's a member of the group, probably the family Esocidae, but when we get into the base of the Paleocene, the earliest locality in the Paleocene we have something we can put in the genus Esox. And there's also other evidence when you're looking at the morphological features of change right at the boundary. So this is that Asteriophyson, and the feature I wanted to point out to illustrate that is this opening with a very sharp ridge at top, which is present all through the Cretaceous into the late Maastrichtian. When we get into the Paleocene, the difference is that the opening gets rounded and a wider bar across it. There's also differences in terms of dorsally present. So a morphological jump, we don't know what this means, if it's a new genus, if it's a slight change in one species replacing another, but it's significant that this change is occurring right at the boundary. So we don't know what's happening, whether this is rapid evolution over that time period, or whether it's new taxa coming in. But whatever it is, there's change at the boundary. And there's also change in terms of diversity. Taking that morphotype approach to looking at diversity based on centra, these are the raw numbers. 20 teleosts in the Hell Creek Formation, 8 in the very base of the early Paleocene, Porkin 1, and then slightly later time it moves up to 12. But again, there's a sample size effect, and we can reduce that sample size effect by looking at number of taxa per locality. So again, a jump going from Dinosaur Park Formation into the Hell Creek, into the Hell Creek, and then across the boundary, a drop to about 7 per locality, and considering we've only got about 3 localities from that time period, that's reflecting a very low diversity in the total assemblage. The number of localities increases in the Puercan 2-3, but when you and the total number of taxa we know from that time period is higher, but when you average it per locality, it stays at this very low size. So it looks like that indeed the fish were hit pretty hard by whatever happened at the boundary, and their recovery was slow. Again, acanthamorphs strikingly maintain a very high abundance within the assemblages, actually increasing to so that typically more than 70% of the centra that we get in an assemblage are a member of this acanthamorph group. So that this is reflecting total diversity. And so just in summary, what I've been trying to do is look at fish to look at patterns of change throughout the late Cretaceous and try and understand that aspect of the assemblage. It helps understanding the big picture because whatever is happening to the fish, other things would be affected too by especially aquatic things like turtles and crocodiles, but because they have different physiologies, might be responding in different ways. But one of the things that's clear is that there's a couple of very strong faunal changes in the late Cretaceous, one between the Cenomanian and Turonian, and one about the time of Dinosaur Park. terms of fish assemblages, these early fish assemblages, teleosts, are diverse but dominated by fish of small size. In the late Maastrichtian, there's an increase of complexity of the communities. The diversity, average diversity of a locality is increased, and you get more teleosts of larger size. And that there is a big change going across the boundary. There's some new things that appear, and there's low-level taxonomic changes in lineages, as well as a decrease in diversity, although acanthamorphs remain the dominant group of fish. So it leads to a number of questions, and it gives us a different way of looking at what the events are at the boundary. And one of the big questions, given these rapid changes, are we dealing with taxa moving in from another locality, or are we dealing with rapid evolutionary change over a short period of time? And there's a number of ways we can approach this, but the big one is looking for more material, looking for more samples, and trying to get more data. So with that, I just have to acknowledge a large number of people, including my co-authors in this study, especially Andy Newman and Mike Newbury and Jeff Eaton for providing material and helping interpret the material. And with that, thank you for listening.